You're welcome to this session, and we want to discuss about DNA transcription. In the previous video, we looked at DNA replication, and we saw that DNA replication, which happens in 3 steps, that is initiation, elongation, and termination. We saw that there are even there are drugs or inhibitors that we can use to inhibit replication, whereby the inhibitors include fluoroquinolones, and these fluoroquinolones like ciprofloxacin, levofloxacin, these are being used to treat UTIs, and they act by inhibiting DNA topoisomerase. They inhibit topoisomerase, which we saw it is a bulldozer that inhibits this from relieving the supercoils. So, and another one we are being utilized is the nucleoside reverse transcriptase inhibitors. These are antivirals like stavudine, we have abacavir, we have tenofovir, the adenosine. All these drugs, for them, they inhibit reverse transcriptase enzyme by incorporating a dideoxynucleotide. Then there is also anti-cancer drugs like gemcitabine, and citalabine. They are also being utilized, and these guys inhibit DNA polymerase enzyme. So these are the drugs that we can use to inhibit DNA replication. So in today's video, we want to look at DNA transcription, which follows replication. And please always remember to subscribe. If you haven't subscribed, hit the like button and keep sharing your comments. So in DNA transcription, we want to see how to, when we have a double stranded DNA, how we use it to form RNA. So we are forming single-stranded RNA from double-stranded DNA. And how is that we are copying information from double-stranded DNA. We are putting it into a single-stranded RNA, to be used up as a precursor in protein or to direct protein synthesis. So this is what we call transcription. Transcription, you are forming a single-stranded RNA from a double-stranded DNA, and we are copying information from a double-stranded DNA helix into a single-stranded RNA to be used as a precursor in or to direct protein synthesis. That is what we call transcription, which is part of the central dogma. And this transcription uses a major enzyme known as DNA-dependent RNA polymerase in both eukaryotes and prokaryotes. And we see that in the prokaryotes, if we have prokaryotes versus eukaryotes, we're going to see in prokaryotes, it is known as RNA polymerase. This RNA polymerase in prokaryotes, it has 2 alpha subunits and 2 beta subunits, and these are the 4 subunits, and the 5th, it is called sigma subunit. So here we have the beta subunit, 2 beta subunit, and 2 alpha subunit, and these ones, the 5th one, so these 4 make what we call the core enzyme. This is the core enzyme. Then the sigma subunit, for it is the 5th subunit, which binds to the promoter. So what makes the DNA holoenzyme, the RNA polymerase holoenzyme to recognize the promoter region is the sigma subunit. Sigma subunit is the one that binds to the promoter region to initiate transcription. So the whole of them, the sigma subunit together with the 4 subunit, will form a holoenzyme. So in prokaryotes, it is one enzyme, is one holoenzyme made up of 4 subunits, that is 2 alpha, 2 beta, together with the 5th subunit, which are calling sigma subunit. And these are the ones that participate in transcription, that is in prokaryotes. Then in eukaryotes, we are going to see that RNA polymerase utilizes, we have RNA polymerase type 1, type 2, and type 3. Whereby type 1 is for formation of ribosomal RNA. This is for messenger RNA plus small nuclear ribosomal RNA. And then this one is for transfer RNA. This is in eukaryotes or higher animals. In prokaryotes, it is the same enzyme. Then as we continue, we are going to look at the promoter region. The promoter regions in this, we are going to also differing depending in the prokaryotes and eukaryotes. In the prokaryotes, the promoter region, it includes the tata, which is always -10 base pair units upstream, and we have also the -35 base pair units upstream. So these are the 2 promoter regions found in prokaryotes. And these are the regions where the RNA polymerase holoenzyme uses the sigma subunit to attach on the DNA template strand to initiate DNA synthesis, to initiate what we call RNA synthesis. Then in the eukaryotes, we're going to find the promoter region as the tata box. If I find the cat box, and the GC box, whereby this is -25 upstream, this is -75 upstream base pair, and this is -90 upstream. Upstream means it is before the start point. If the start point is here, we're going to see the promoter regions are base pair units upstream. Then when from the start point is downstream. That's what we are doing. So we have seen also these are the promoter regions, both in prokaryotes and eukaryotes, whereby we have the tata box, the cat box, and the GC. So after seeing these differences between prokaryotes and eukaryotes, others are going to be the same. They're going to be the same. And this DNA has what we call the sense strand from 5' end to 3' end, and this one we call it the coding strand. And then it has also the DNA template, which is from 3' end to 5' end. This is the DNA template, or the template strand, or you can call it antisense. The antisense strand, the coding strand, you can call it sense strand. So those are the strands. So the template strand, which is from 3' end to 5' end, is the one that we utilize for synthesis of our RNA. So we shall see that our holoenzyme comes using the sigma subunit, attaches on the promoter region together with the 4 core enzyme, that is 2 beta and 2 alpha, and it attaches on the promoter region using the sigma subunit. And after attaching on the promoter region, this one might be the -10, what we call tata, TATA-AAT. So this one, if it is in prokaryotes, we see this promoter region, the RNA polymerase come and binds. RNA polymerase or enzyme binds, and after binding on the promoter region, it begins moving along the template strand. And as it is moving along the template strand, when it reaches on the start point, it will begin synthesizing. So we are going to see here that the sigma subunit together with the whole enzyme, what happens in this, as we normally tell you that these reactions occur in stages, whereby the stages we have the steps. We have initiation, we have elongation, and we have termination. So initiation starts by the RNA polymerase binding to the promoter region, binds to the promoter using sigma subunit, then it starts moving along the DNA template strand. And this, when it binds, it has two functions. One, when it binds, it unwinds the double-stranded DNA. After unwinding, it adds two ribonucleotides. After adding two ribonucleotides, we shall see the first formation of a first phosphodiester bond. It leads to the formation of a phosphodiester bond. So in this process, we do not have the RNA primer. The RNA polymerase or enzyme itself, it has the ribonucleotides. So it keeps coming to add, whereby it initiates by adding the first two ribonucleotides, using ATP, using UTP, using CTP, and GTP. So it adds the first two ribonucleotides and facilitates the formation of the first phosphodiester bond. And this is the initiation step. This one, don't forget, is occurring on the template strand, which is from 3' end to 5' end. Then the coding is from 5' end to 3' end. So after it has initiated, the first step is it moves along until it reaches the start point at positive number 1. Then it moves downstream and adds two first ribonucleotides and facilitates the formation of phosphodiester bond, which are going to be the precursor for elongation step. So in summary, we are saying after the RNA polymerase binds on the template strand, it does two roles. First, it replicates the DNA. Then the second, it adds the first two ribonucleotides and leads to the formation of the first phosphodiester bond. And that is what initiates DNA transcription. And that is the initiation step. Then when we go to elongation, elongation is that this DNA polymerase continue to move. So we know that when it is moving and it reaches 10 phosphodiester bond, 10 phosphodiester bonds, it breaks away. The sigma subunit breaks away. We see sigma subunit breaking away, and as it is moving along these strands, it keeps adding complementary base pairs. So we are going to see if this was the first phosphodiester bond, it's going to keep reading the base pairs on the template strand, adding the complementary base pairs. It continues to move along with its core, the core enzyme, remember. Because the sigma subunit breaks away after 10 phosphodiester bond. So the core subunit continues to move along the template strand. We are having the core subunit, the core enzyme without the sigma, and it continues moving, synthesizing our RNA, adding complementary base pairs. And this one, elongation step, it is utilizing UTP, it is utilizing ATP, so it is using ribonucleotide triphosphates, including GTP, including CTP, adenine triphosphate, uridine triphosphate, guanosine triphosphate, and cytosine triphosphate. So it keeps adding these ribonucleotide triphosphates by reading the bases on the DNA template, adding the complementary base pair, and it continues moving along until it reaches what we call termination signal. When it reaches on the termination signal, it stops there. Once it detects the termination signal, the RNA polymerase does not continue to move. And what happens now? It enters into what we call termination step. But under elongation, what you have to know under elongation is that the RNA polymerase continues to move along the template strand, adding complementary base pairs using ATP, UTP, GTP, and CTP to add complementary base pairs along the template strand. And we have said, as it is moving, when it forms 10-phosphodiester bond, the sigma subunit breaks away to leave the core enzyme. And the core enzyme continues to move, adding more complementary base pairs until it senses the termination signal. And this is when it will stop the elongation step and we enter into termination step. And this termination step occurs in two ways. And allow me to put them here. The termination step is occurring in two ways. Whereby termination, the first one is low factor dependent, and we have also the second one, which is low factor independent. Low factor independent. In low factor dependent, here we use the low factor. And this low factor contains ATP dependent helicase enzyme. So this ATP dependent helicase, we are going to see it comes to the termination signal, it adds the low factor, and this low factor having ATP dependent helicase, it breaks away or it displaces the core enzyme. And when it removes the core enzyme of the DNA of the RNA polymerase, it terminates the chain. So we see here we have already terminated in the presence of low factor. Low factor utilizing helicase, which depends on the ATP to displace or to remove the core enzyme. That is mechanism number one of termination. And mechanism number two, in this termination step, we are going to see that this low independent, we see the newly synthesized strand of RNA forming what we call a hairpin loop. Forms a hairpin loop which ends with 4 units of uracil. This is a hairpin loop. It forms a hairpin loop. And this hairpin loop formed is the one that terminates the elongation step. It is terminated at that point after 4 molecules of uracil. But after forming this hairpin loop, this newly synthesized RNA strand, comes and forms a hairpin loop ending with 4 molecules of uracil. And when it is formed, it initiates the termination of the elongation step. So we have seen termination is in 2 mechanisms, whereby we have low factor dependent and low factor independent. The low factor dependent, it is utilizing ATP dependent helicase enzyme, which displaces the RNA core unit from the strand, leading to the termination of the temp of the RNA. Remember, synthesis is occurring from 5' end to 3' end. Even here we have 5' end, 3' end. Here we had 3' end, 5 as a template. So we are forming the RNA from 5' to 3', using a template which is from 3' end to 5' end. And this one marks the end of transcription, whereby in summary we have seen different steps, and we have seen the differences between eukaryotes and prokaryotes, whereby in prokaryotes we are using the holoenzyme made up of 4 subunits, 2 alpha, 2 beta, and sigma subunit. And we have seen in eukaryotes we have RNA type 1, type 2, and type 3, whereby type 1 is for ribosomal RNA, then this is for messenger and small nuclear, and this is for transfer, which is type 3. And we have seen also the defined promoter regions, whereby we have the 10 region, the -10 and the -35 upstream for prokaryotes, and the eukaryotes, they have the Tata box, the CAT box, and the GC box. And those are the differences between prokaryotes and eukaryotes. And we have seen the steps that involve in replication, whereby we have the initiation, elongation, and termination. And we have seen all of them, what they do. Please do not forget the function of sigma subunit in prokaryotes. It is the one that recognizes and binds the promoter regions before initiating transcription. And do not forget that we do not use RNA primer in transcription, whereby the RNA polymerase has its own nucleotides. So it adds the first 2, leading to formation of the phosphodiester bond, and it keeps adding more during elongation step. And we said when we form 10 phosphodiester bonds, then the sigma subunit breaks away, and the RNA core enzyme is the one that continues to add complementary base pairs on the template strand using UTP, ADP, ATP, GTP, and CTP, which are ribonucleotide triphosphates, elongating this. And we don't forget that the synthesis occurs on the template, or what we call the antisense strand. That is where it occurs, which is from 3' end to 5' end. And we have seen that if it continues, it reaches the termination signal, whereby the RNA core enzyme stops there, and it is terminated by 2 mechanisms, which is rho dependent and rho independent. And whereby we have said rho dependent is using ATP associated helicase, which displaces away the core, and in independent, we have seen the formation of a hairpin loop ending with 4 units of uracil. Thank you so much. In the next video, we shall look at transcription modification or processing, and we shall look at also inhibitors of transcription and the genetic code. Thank you so much for watching until the end. Always be there. Remember to subscribe, remember to like the video, and share your comments. Thank you so much.